So, good morning, everyone. So, today's lecture will be a little bit late. We will start at 11:15, but uh, means if someone having any doubts regarding our software or our installing any problem they are facing, they can ask. All the queries will be addressed by Dr. Kapil Chaudhary. So, if anyone having any problem related to R, they can ask. किसी का कोई डाउट नहीं है? Good. So still participants are joining now. So remember that if you participate actively, then only we will provide you certificate. I know some of us here for certificate, but we will provide certificate only if you means seriously take all the sessions.
Okay, morning participants. So till our today's speaker join us. We will start our last from correlation regression to design of experiment. Okay. So in last class we studied about the basic descriptive statistics, how do we calculate the mean, median, mode, and other thing which are very essential to start the data analysis. So first of all, today we will first of all discuss the correlation regression, and after that we will proceed for basic design of experiment, how we go for design of experiment, what are different design of experiment, and what is analysis, how we conduct different design, and how their analysis we can carry out in our software. Okay. So first of all, let me share my screen. Okay. One second, one second. It is asking when you share the manual of this workshop. Only they will provide after completion of this training. Vijay, whether my screen is visible? Visible. Okay. So in last class, we discussed about basic descriptive, and today we will discuss about the basic correlation regression and one or two design approximate. Okay. So before start, first of all, you have to install some package to run the correlation or regression. These are packages, stats and forecast. Once you install these packages, then after you have to import in your R directory. That for that you have to run library and different library for maybe for stats and forecast. Okay. So once you run all these basic codes and library, then we will proceed further. So first of all, you have to import your data. As in previous class, I told you that there are different methods. Either you can go for the directly environment option. Here is either from here we can import our data, either from maybe Excel or text file. Okay. Another way you use any command is there. Suppose I'm using here core regression. It is my data is there, and from this I want to import file. Okay. So if I run this, then it asking where is the location here. Okay. Suppose my location is here. It is my data. Okay. So if I want to see how my data is, so it consists four different variables. First is pp, that is plant population, average plant population is there. Second is plant height. Third is number of green leaves, and fourth is yield. Okay, so they have taken 46 different sample data and from different plot. Okay, and from each plot they have taken four variables. So that is pp, ph, ngl, and yield. Okay. Now, if I want to find out a correlation between each pair of the among the PP, PH, NGL, and YLD, then for it, I have to run core regression. Core regression is my data. Okay. If again, if you want to see your head and tail of data, then you have to run it head core regression. Core regression is my data. Okay. So it will be my head. In head, it will show you above 6 rows. Okay. And if I want to see tail of my data, tail of my data, that is core regression, then sorry, core regression. Okay, so it is tail of my data. My data consists already, I told you, 46 and data point. So here the last 6 rows are consist 41 to 46. Okay, with their heading of the variable. So again, if you want to directly run correlation, means it will be show you correlation among all these four variables. Okay, so for that you have to run this command core regression. Okay, so if I run it, then it will show you how the correlation reflect between different variables. Like first PP and PP, they directly correlated. So that's why the correlation is perfectly positively correlation. That is why it is one. And if I want to see the correlation between PP and pH, then it is 0.23, is positively correlated. So here, as I can see, all variables are positively correlated. Positively correlation means it, if one variable's direction goes up, then other variable's direction also goes up. So it is the basic. Vijay, I, Amarkant sir, I think so. Rest of carry on.
Okay, Amarkant sir, join now.
Okay, we will start rest part in other classes.
Good
Sometime we will
morning,
get.
sir. Sir, unmute yourself. Sir, please unmute yourself.
Okay, sorry for the delay. Actually, RAC members are here, so there was meeting going on.
Yes, sir.
So I asked for the excuse and okay.
Okay, sir, no issues. So, uh, good morning to everyone. So, today we are fortunate to have Dr. Amarkant Kushwah, sir. Actually, currently he is posted at CISH, Lucknow, and today he will deliver the lecture on advanced techniques in plant breeding data analysis. So, Amarkant, sir, you can continue.
Okay, thank you, Vijay.
Sir, your video is stuck. Hello, sir. Actually, he is having some internet issue, so shortly he will join again.
Hello, am I audible?
Yes, sir. Yes, sir. Yes, sir.
Oh,
I'm making new course. 1 second. Sir. Yeah, sir. You can continue, sir.
Okay, thank you, Kapil and Vijay, for providing me opportunity to interact with these guys and tell them about what is QTL, what is genomic selection, and what is going on around in plant breeding. So, कैसे शेयर करना है? स्क्रीन शेयर करनी है?
हाँ सर, स्क्रीन शेयर कीजिए।
एडवांस
ये शेयर स्क्रीन का ऑप्शन आ रहा होगा सर।
हाँ, शेयर स्क्रीन।
तो गो फॉर एंटायर स्क्रीन।
दिख रहा है? इज इट विजिबल?
ठीक है सर।
यस सर,
विजिबल सर।
विजुअल सर।
अभी फुल स्क्रीन मोड में है कि वैसे ही है आपने?
हाँ सर, फुल
फुल
स्क्रीन।
स्क्रीन है।
हाँ, ठीक है। So friends, today I'll be dealing with the QTL mapping strategies and basics of genomic selection. Not I am going into deep into the models and all, and I will tell you how to proceed with genomic select aspect. And QTL mapping strategy, I will tell you about what are the different softwares, and I will be dealing with ICI mapping, which is the most advanced and sophisticated one. So before going into directly into how it is done, let's delve into the basics of the QTL mapping. So, as we all know about the traits. There are qualitative traits and quantitative traits, and there are different. These two are defined based on their features. So we know that qualitative traits are governed by one or few genes. They have major contrasting effect on the features, and there is no major influence of the environment on the external appearance, means phenotype, and they show discrete variation, and they are study based on the ratios. And these are the most fundamental before we go into further. And quantitative trait, as we know that these are governed by many genes which have cumulative effects on the phenotype, and but all those genes have minor effect, means say individually their effect can be very minor, so that it cannot be quantified on the phenotype, but cumulatively they are producing the phenotype, and they are under the high influence of environment, and they show continuous variation which range from one extreme to the other extreme, and the mean lie in the uh, major chunk lie in the mean area. Okay, and it show continuous variation, and higher order statistics is required, like standard deviation, variance. We all know about these things, but before going into I think it is better to. So what is quantitative trait loci? So as we seen, what are quantitative traits? These are means there may be some there are factors which are there may be some factors which are responsible for the manifestation of those traits, like by definition, the quantitative trait loci are the genomic region in genome which is responsible for the expression of the quantitative trait. So, and these QTL region may be defined to one location in the genome, or maybe there are many QTLs responsible for the trait of interest. Like for here, I have presented one of the QTL which I have studied in my PhD program, and this QTL is responsible for Bakani resistance in rice. So when the mapping was done, it was found that there are four QTLs responsible for means quantitative trait locus, and four QTLs were responsible for the trait of interest, means Bakan resistance. So it is not necessary that whenever you go for quantitative trait loci mapping, you will get only one QTL. You may get more than one or two QTL also. And moreover, when you delineate those QTLs, you may find many of genes lying within that region, and you have to pinpoint which gene is responsible within that QTL, which is causing the trait of variation in the trait of interest. And these are the whenever you go for QTL mapping analysis, QTL mapping studies, load values are considered. R2 means R square value, which says that how much variation is explained, how much phenotypic variation is explained by that QTL. And additive variance means if that QTL is having additive variance, means it can respond to the selection and it can be utilized in the breeding program also. So our means why we go for mapping, as we already know that gene until unless we know where are those genes, how can we utilize in those genes or QTLs in the breeding program. So we have to map to effectively use those things in our those QTLs or genes in our breeding program. That's why we go for mapping QTL or any gene for per se. So basics of the QTL mapping is before going into QTL mapping directly, you should know what is linkage mapping. Linkage mapping is the order of gene. Like here it is depicted A, B, C, D, and E, and it is linear in order and distance between them, which is represented in terms of recombination frequency between those two genes or markers. This is not just a gene; you can say marker also. And this linkage map or genetic map is constructed based on point of means when you go for classical genetic experiments, you find 2-point analysis, 3-point analysis, multi-point analysis, and these are the basics for means based on the recombinations between those genes. You can find recombinants, parental types, and the frequency based on the frequency of those recombinants out of the total population you have screened. The recombinations can be worked out. When you all might be knowing when you say that gene are linked, when you say that the genes are not linked, means independent segregation. Yeah, it's not about only genes. What means when I'm talking about gene means here I'm talking about also markers. Means when you go for genetic analysis, you are based on recombination frequency. Marker analysis also will give you recombinants, and based on the recombinants, you identify how much recombination is going on within that region between those two markers. And counting those two markers will out of the population will give you the proportion of recombinants, and based on that recombination percentage you will say that these two markers or gene are lying this much distance apart, because the linkage mapping distance is depicted in terms of centimorgans. Okay, for this is the historical aspects. It was the first linkage map which was constructed by A.H. Student in Drosophila, and he mapped I think six genes. The data was provided to him by T.H. Morgan, and he was algebra student at the time, and he took the data and he came out with means algebraic equation and say that these are the recombination frequency, and based on that he has constructed the first linkage map. So linkage map or QTL mapping study based on the recombination. So how you induce recombination? So recombination is induced by creating mapping population. Okay, so mapping population is created. Mapping population is created by controlled crossing. Means controlled crossing in the sense that you are controlling the parents. Parents means you know how much distance is there in between the parents, and whenever you are going for QTL mapping, you select the parents so that one is having the means they are having contrasting feature for the trait which you are going for mapping. So most of the studies you might have gone through for QTL mapping is based on recombinant inbred lines, and other R's like F2, double haploid, near isogenic lines, they all have their own advantages and own means disadvantages. Means it depends on the focus of your objective of your study, how you are going into. So if you are working means if you are you have to go for fine mapping, you have to develop near isogenic lines so that most of the background noise are nullified. And if you are going for preliminary mapping, it means you can go for F2 if you don't have means much resources, much time. and you want to go for preliminary mapping, you can have F2 because it can be generated within less period of time. And but F2, there is problem with F2 that you cannot means replicate them across the environment, or means replicate per se in the same environment also. So hence, RIL is considered as the most suitable mapping population, which many workers are using for QTL mapping studies, because these are the fixed lines, and coming out of the recombinations which have gone through in F1 individuals, and these are the fixed lines. So recombination bins or blocks are there, and these are highly structured population because only two parents are there. You might have gone through Jivas where there is a requirement of unstructured population, and historical recombination is to be taken into consideration. But here we are just countering two parents, so the recombination blocks are coming out of only those two parents, parents A and B. And RILs, one feature of RILs is it is generated through continuous selfing and selecting through SSD methods. And other specialized mapping populations are, means if you are tapping, you want to tap more means recombination from different genetic backgrounds. So you can go for magic development of multi-parent advanced generation intercross population. There you can have, and then you can have nested decision mapping panel also. So these are things for creating a recombination so that mapping can be done. So this is the representative picture how you are going for. So means this is objective oriented. Whatever resources you are having, say for example, one PhD student is going for a mapping study. So is it justifiable to go for means real advanced generation rules, means F10, F8? It is not justifiable until unless he is having shuttle breeding program or rapid generation advancement facilities. But a means highly ambitious breeding program where a lot of resources are there, they can go for means this development of rails and all. So it is highly objective oriented. If you means if you want to generate faster mapping population, you can go for F2. Double if you want to have same information like real, you can also go for double haploid production. But it should be the facility should be available. Means you have to have haploid and user lines and double haploid production facilities. So here I'm showing like duration can be reduced in case of double haploid production lines, and these are the in case of it will produce the same kind of recombination blocks as that in case of the RILs. So this is the first requirement where you have to induce recombination by crossing the parents and generating population. The another requirement is the marker. So an array of markers systems are available right now. Earlier it used to be very limited number of markers, like if you go before 1970, you are they were only morphological markers, and very less mapping studies were also there. Then there come through evolution, means identification of isogyme and allergens. Those were also used. And then DNA based markers came into being. Earlier were hybridization based, where radioactivity was used, like RFLP. Then PCR based random markers like RFED. All those things I think that you have gone through, and you might be knowing all about markers, I think. And the most recently, which is the most recently, the marker system which has been evolved, like SNP and indels. SNP is becoming the most favor means choice of marker, because the cost of sequencing and genotyping is going down day by day, and it is the number of polymorphism when you go for SNP is high as compared to other marker systems. So these are the most favorable ones, and like many crops are having SNP genotyping chips like 80K chips, 50K chips, 3K chips. So they can and the cost using genotyping cost using those chips are very less. Per data point you may encounter about $1 or $3. So that can be counted for having an large mapping population to be genotyped using those SNPs. So this is the basic critical mapping strategies. first step is to phenotypically evaluate and screen the germ plants before making any step further. So in my opinion, the phenotypic evaluation is the most important part. Whenever you go for QTL mapping strategies, your phenotype should be so much accurate and tested in multi-environment, so that you don't have any kind of doubt. Means if you are having ambiguity regarding phenotype, you will land nowhere. So phenotypic evaluation and identification of parents for making any means population, it is very important. You should like for example, you are going for disease resistance, you should screen in so robustly ki there should be no ambiguity, and clear cut clear cut contrast should be there in the parents. Like one is resistance, means it is resistant, is showing clear cut hypersensitive response, and the susceptible one is showing susceptibility. And the scale of means phenotyping should be well defined, because that is where you are going for phenotype, putting the phenotype in the mapping studies. Okay. Then genotyping may require your parental polymorphism and genotyping the population. Parental polymorphism means uh, the parents which we have used for developing mapping population should be genotyped for as many as marker possible, so that uh, because when you go for SR markers genotyping, so it's there's a general rule like 10% polymorphism is observed in case of SR. If you screen random SSR markers through like 1,000 SSR markers, you'll end up to like 100 or 150 polymorphic SSR markers. It is a general connotation. And once you have gone through parental polymorphism, use those markers which are polymorphic between parents for genotyping the populations. Then once you have done your genotyping the population and parental polymorphism, then come this most easiest part, that is statistical analysis and mapping, and then confirmation and validation. This means general doubt like what is confirmation and validation. Confirmation means when you have mapped the QTL, and again you are confirming by genotyping the same population using the same marker system, and saying that whatever QTL has been mapped is again coming to that location. So this is kind of confirmation. Validation means you are testing those QTL which you have mapped in other mapping populations. Means not in the population where you have used the P1 and P2, but other like P3, P4, P5, P6. Whether that QTL means resistance is governed by the same QTL or not. Okay. It is not necessary that when you go for validation in the resistant lines, other than the resistant line you used for making population will be the same, because there are many other means if you are selecting lines from different ecological niche, they might be harboring different QTL for that resistance, because the race available in that niche is very different from the race you have used for mapping your QTL. So these are the statistics like single marker analysis, interval mapping, composite interval mapping, multiple interval mapping. Means whenever you are going for QTL mapping analysis, step by step you should go further. Means whenever you are I'm talking about statistics. First you have to go for single marker analysis, putting one markers and the phenotype classes, and see whether these phenotypic classes are showing difference. Means the difference between phenotypic classes and the difference between the genotypic classes correlating with each other or not. So once you find that these markers are showing single marker analysis, you are getting the QTL. Then if further proceed for means more robust models like composite interval mapping and inclusive composite interval mapping and multiple interval mapping. So why you go for multiple interval mapping? So the simple logic is that when you use this simple interval mapping, single marker analysis, you are having one marker and one QTL. So whenever you go for pyramiding or marker system selection program, the chances of getting false positive is high. Say for example, you are having a marker and QTL interval around about 10 centimeter, 10 centimorgan apart. So out of 100, you are having 10% chance of recombination. So the marker genotype will be positive, but you are not having any QTL. But if you bracket bracket the QTL with another marker like M1 and M2, so and both say for example R1 and R2 is 10 centimorgan. So what is the probability of getting a false positive in that case? 1 by 10 means 1 by if 1% recombination is going on, 1 by 100 into 1 by 100, that is 1 by 10,000. Means out of 10,000 you are getting one individual like which is false positive. So it is a customary like you should bracket your QTL with the two markers, one in left and one in right. Yesterday I saw that someone was asking about bulk segregant analysis, and it's means so this can be also utilized for mapping QTL. So this the principle behind the bulk segregant analysis. Bulk segregant analysis is you are having you are developing mapping population or any say for example disease resistance. You say P1 P2 you cross, you are getting F1 and then F2. So if you are your resources are not permitting you for mapping, means genotyping the whole population, or you want to go for preliminary screening whether the gene is there or not, or your the available marker is available with you, can means any of them is having significant association with the trait or not, means co-segregation is there or not. So you can go for bulk segregant analysis. This is very simple technique, and it doesn't require any means full population genotyping. Here what you have to do is generate mapping population like F2, and you phenotype the whole F2 population, and you take 10 extreme bulks, means 10 from one extreme and 10 from another extreme. So and then bulk those extreme and genotype them with your markers, and see which markers are showing, which markers is showing which marker is co-segregating with the trait of interest. So this is the formula which you should take into consideration. Erroneous linkage की probability क्या होती है? This can be taken into consideration. That's why this 10 number came, like people ask why they are taking 10. So when 10 is taken, so n becomes so means so large that 1 minus 1 become means this become 0, p become 0, pfl become 0. So based on this bulk segregant analysis, one technique that QTL came into being by Takagi et al. In 2013. So bulk segregant analysis was for means proposed for qualitative trait, like Michel Moore has reported for downy mildew resistance in lettuce. So it was thought that whether we can utilize this technique for mapping QTLs or not. So earlier the number of marker system were not that much robust, and number of marker limitation of marker numbers was very prominent. So when with the advent of sequencing platform and genotyping platform, it become easy for in crop like rice. So first report was in case of rice, where the mapping population was developed like F2, and lowest bulk they have, I think, map for sheath blight resistance, and the bulk for lowest resistance and highest resistance, and they map this. Okay, they sequence this region. They sequence this bulk, and they sequence this bulk to an optimal depth, and align those reads on the reference genome, and identify where the SNPs are means at that particular at which particular site SNPs are there. So based on this, they have calculated the SNP index. Like in reference genome, you see here reference genome C. So out of 10 reads, you are getting 4 SNP, means which is not C. So the SNP index is 0.4. Similarly, at other location, you are getting 10 out of 10 SNP, means different from the reference sequence, means SNP index is 1. So when you sequence lowest bulk and highest bulk, and you calculate separately SNP index in those two bulks, and after that calculating SNP index in highest bulk and lowest bulk, you get a delta SNP by a formula. Like SNP index here, SNP index delta SNP index is calculated as SNP index of R bulk, means highest bulk, minus SNP index of S bulk. So if SNP index is 1, that indicates region is harboring the gene responsible for mutant phenotype, or QTL harboring the QTL. So these SNP indices are scanned across the genome, and chromosome wise, it gives a result chromosome wise, and you can delineate the region where the QTL is harbored. So they map this means pyrocluria or IG. I think they have mapped this gene, this QTL, to a region 2.39 to 4.39 on chromosome number 6. Okay, so this is the QTL-seq, and regarding practical analysis of QTL mapping, there are many platforms available for QTL mapping, but these three are majorly used, like QTL ICI mapping, which is developed by Chinese Academy of Agricultural Sciences, and QTL Grapher, that is North Carolina State University, developed by North Carolina State University, and R-based packages are also available for QTL mapping. So I think ICI mapping is the most recent one. This was most recent and most robust, means these means having lots of model for different different practice, means different calculations and mapping, and it can map populations of different kind of population can be utilized for mapping by using ICI mapping. So I'm here I'll show how to use ICI mapping for QTL mapping studies. So this was developed by Mangaton 2015. So this take into consideration different kind of populations, means whether you are having DH, you are having real F2, back cross F2, anything. You can have different things, different population. You can go anything, means you can go for. And earlier, like with QTL cartographer, you have to create a linkage map by using map maker. But in case of QTL ICI mapping, you can you can develop a linkage map. You can create a linkage map in the software itself. Okay, so this is the practical exercise of ICI mapping. Is there any query, Vijay, so far? Hello?
Yes, sir.
No, sir. No, sir.
Okay.
If anyone having any regarding this lecture, they can ask.
So this is the data preparation. People, means students especially, get stuck at this point. Sir, how to create the file for mapping? How to create means how to put genotyping file, like that. It is very easy. You download the software actually. Yeah, okay, I will not provide the link. Okay, which I will provide the link for downloading this software in the manual for manual. Okay.
Yes, sir. Yes. Okay.
So this is the method for data preparation. For when you are having when you are going for mapping QTL mapping, and you don't have linkage map for that. Means even that you don't have this anchor chromosome anchoring. So in that case also you can use this ICI mapping. So here in this case, I assume that you know the chromosomal location, and you want to develop a linkage map. So for that map function is being utilized, and the file is created like this. Your this can be created in XLSX format, and these are having 3 tabs like general info, genotype, and anchor. General info will tell you 7 mapping population means I think it is F2, and mapping function is second option is mapping function. Mapping function may be Kosambi, Halden, or Morgan. So mapping function is nothing but to encounter the anomaly of means this double crossover and interference. In Halden mapping function, in Morgan mapping function, the one which was earlier means the first one that student used, he didn't encounter any double crossovers, means between two gene pairs, one gene pair, sorry, two genes. So for that, Haldane has proposed that maybe there are chances that double crossing over may happen, and you are getting parental type only for that matter. Double crossover will lead to parental types, okay? And but though crossing over is there, recombination is going on, but you are getting parental type, so that recombination frequency is going down, okay? So for accounting for that, Halden has given means his mapping function that can account for double crossing over. Accordingly, there is also a second anomaly, that is interference. One crossing event of one crossing over may interfere the formation of another crossover events in the vicinity region. So that should be accounted for as well. So in most of the cases, Kosambi mapping function is being used for mapping. Second is marker space type, whether you are you are taking into consideration two markers. Between two markers you are taking one interval, or you are means you are putting two markers cell. So it is a way of representation, whether you are representing interval or positions. It is better to go for position, because you want to have QTL within that interval. You don't want to have QTL interval between intervals, because that will create the problem in future. So it is better to go for position. And another column, another row is the number of markers, how many markers you have used for genotyping the population. And the next one is size of population. Accordingly, you have to create the file. Okay, in one Excel, you have to create 3 sheets. One is general information, another is genotype, and third is your anchor. Which means in one column, in anchor sheet, you are having in one column name of marker, and another is in B, you are having number of on which chromosome it is located. Okay. Then when you are going for linkage mapping, the load value is considered, and based on that recombination and load value, it will create your linkage map. This linkage map information will be required for further QTL analysis. Okay. So BIP function is BIP function is utilized for mapping QTL. It means biparental population, because in QTL mapping mostly utilize biparental mapping population, so they have given this BIP function there. So in data preparation for BIP function, it is having 5 tabs: general info, chromosome. Chromosome is this in this chromosome wise, you have to put on chromosome number 1 how many markers are there, chromosome number 2 how many markers there, chromosome 3 how many markers, likewise. Okay. And linkage map, whatever the results you are getting from here, means linkage mapping, you have to put that linkage mapping results here in the third tab. Okay. and then genotyping file and genotyping data and phenotyping data. One point to be taken care here, like whenever you are in the say for example, column wise there are markers and row wise it is genotype. So whenever you are putting genotyping data, whatever the genotype wise you have put from B to n number of rows, it should be same in the phenotyping file. Otherwise, it will create problem and ambiguity will be there in the result. So it should be there should be coherency between genotyping data the individuals in the genotyping data and individuals in the phenotyping data. Okay. So this is the notation for creating genotyping file. So either way you can go, either by coding by number or coding by letter, but not mixed. Okay. So when codominant marker is considered, 2 is considered as high parent, 0 is considered as low parent, and F1 is considered as 1. Okay. So if you are going for letter A, B, H, and if any missing is there, it should be in -1. Okay. And in case of dominant, parents can be identified like in dominant marker. If your P1 is showing your band, it will be written as 2. Parent B, where no band is there, 0, and F1 will be 1. But when this is the P1, P2, and F1, but when you go for the after means F2 or real, so you cannot differentiate between the P1 and H. So both are considered as 1, 2, and type means B type will be 0. So this actually this software allows you to do multiple means you can have analysis of codominant and dominant marker analysis simultaneously. You can, if you are genotyping only with codominant markers, you can go with that, and dominant marker as well, you can go with that. But if you want to combine those two marker system in a single genotyping file, you can go means in a one go. So if you want to have a demonstration, I can show you how it is being done. Slide is my screen is visible, Vijay?
Visible, sir. Visible, sir.
So this is a QTL ICM mapping. So here you see how many functions are there.
Sir,
AOV.
actually, it's stuck down your PPT.
अच्छा PPT दिख रहा है क्या?
हाँ, शायद आपने PPT शेयर किया होगा, आपकी स्क्रीन शेयर नहीं किया आपने।
कैसे होगा यार ये?
शेयर स्क्रीन पे ही सर, या तो आपने सिर्फ वो पर्टिकुलर सॉफ्टवेयर ही शेयर किया है। आप एक बार इसको क्लोज करके फिर से मतलब आपकी जो विंडो स्क्रीन है वो शेयर करो।
बेसिक्स में ही होता है ना?
हाँ, जस्ट ऐसे शेयर स्क्रीन पे जब जाओगे ना आप, तो यहाँ पे जैसे स्क्रीन शेयर या फिर
अच्छा
आपके
अच्छा स्क्रीन
पर्टिकुलर
शेयर,
पीपीटी, हाँ बस वो।
ठीक। और अभी विजिबल है?
हाँ हाँ सर, नाउ इट्स विजिबल।
So this is the software. रुक जाओ, ठीक है। So you can see there are 10 functionalities. ANOVA for multi-environment trials. It can generate multi-environment testing, and ANOVA for phenotypic file. SNP genotype conversion. If you are having SNP genotyping, if you are having VCF format, you can convert it to HAPMAP format like that. Means conversion from AG 80 to 10 like that. Okay, binning of redundant marker. Like if you don't have any anchoring of markers like chromosome wise, you can bin those redundant markers based on recombination frequency and use those information for mapping studies. Mapping function for linkage mapping construction, consensus map construction, mapping of segregating distortion loci. So this should be considered beforehand. Like you have to check. I showed you single marker analysis, no? So why you go for a single marker analysis before going to into QTL mapping? Single marker analysis will give you result, means give you information regarding whether your marker is showing segregation distortion or not. Anyone anyone who can say can tell why segregation distortion happens in comment section? Anyone? So it is general rule that you should avoid those markers which are showing segregation distortion. Segregation distortion arises due to means gametic favorism, means if one gamete is in more numbers, so it reduces the other gamete which is coming from the recombination point. That's why it is considered that it should be avoided. Uh, the marker which is showing segregation distortion, it should be avoided. Then BIP function, which is shows which is used for QTL mapping. Okay, so for that matter, let's start. So for QTL mapping, first you have to go to file, new projects, and you have to give a name to the project like tutorial, and location you can give any location. Sorry. Okay. So could you give a path to your project like it? Here I have given tutorial. Okay, let's see. Let's create another file. Tutorial. So here all the functions are here. Out of these functions, what you have to do, whatever you have to do, you have to select that. Like for first, we have to create a linkage map. So accordingly, we have selected the linkage map. And if you want to do any analysis, means you can, yeah, the software will provide you a lot of means example files also. You can use any of the means example file and utilize those for means for your exercise purposes. And whenever you go for your mapping studies, accordingly you prepare your file and use the same thing. So you have put your data in the XLSX format. I am taking here RISE F2 example. So in the left hand panel, it is showing this is the file is having. So file you in the file you don't have given these are all these information. This is the software itself has calculated all these things. what is the probability, means what is chi-square value, what is the nature of the marker, whether it's codominant or not. So accordingly, the software do all these things. So what is chi-square? What does chi-square say? Chi-square say whether it is following your Mendelian segregation ratio or not. Okay, this can be means tested based on the p-value. If p-value is less than 0.05, means that marker can be avoided for analysis, because it is due to it is showing segregation distortion. So this can be avoided, like it is having less than 0.05 value. This can be avoided. So accordingly, you have to go for all those things before before using all this data into QTL mapping, actual QTL mapping study. Okay, these all marker have been here. This is your anchoring, like chromosome 1. Anchor 1 means your chromosome 1. Okay, chromosome 1 is having this many markers. Chromosome 2 is having this many markers. Chromosome 3 is having these many markers, likewise. So what you have to do, what you have to go for? Threshold value by load. Load threshold is 3.0. You have to group them. So it is doing grouping. So it is grouped based on your load 3 value, based on recombination among genotyping. Now let these as default, because it is it means it serve purpose for different means if you are having some specific analysis, then you can go disturb this. Otherwise, let it be means default, and it will order your marker accordingly based on the threshold value and load. Order it. Okay, this is order. And now based on load, you ripple. Ripple means how many times you are putting means changing one marker with all of those markers. So it will give you means this will give you rippling means one marker, you are marker wise, one marker with all those markers, another marker with all those markers. So you ripple those. So it will create means it will give you robustness about your mapping, means linkage mapping. So based on outputting load score, recombination frequency, and pairwise distance output. So the thing is done here. So all of these files have been created. You can see by double clicking on this. This is the summary file. So here you are concerned with BIF file. This is the important one, which from where you have to extract the information for QTL mapping. So this is having all the information which you have provided for the analysis. Like it convert itself. Like if you have given AB, sorry 120 like that, it converts for its sake X, H, A, and B. You can use X, H, A, and B or 120 or -1 form. Any way you can use. But this convert for its analysis A, B, H, and H into H and X. Okay. You use this information for like here you see for QTL mapping file, you have to have this linkage map. Okay, this one, third one, 1, 2, 3, third sheet. So from where this thing come? So this thing can be created from here. Otherwise, when earlier ICM mapping was not there. QTL cartographer used to be the software which people use for QTL mapping. For that, another software like MapMaker was there, which was used to generate such kind of information. Okay, so here itself means the same software can do that analysis, and you can copy from by selecting this. Sorry. Proceed and put it in Excel file. So and paste it here. So this will sorry, paste like if it will same cell. So text import fixed width delimited, next, finish. So now it is in different cells. Okay, now this can be your third sheet. Can be your third sheet of linkage map. So till this everything is clear. Now your actual QTL mapping, actual QTL mapping. Okay. Now open. Not here. So this is till your uh, linkage mapping, linkage map construction. Okay. Now you have created accordingly QTL mapping file where you are having general info, marker information, linkage map, genotyping and phenotyping file. Okay, let's see. File, new project. Yes. Okay. Now you have to select function BIP, QTL mapping in bioparental population. Now you change this to XLSX and rise up to open. This is the information available in the BIP file. Okay, chromosome first cell, sorry, first sheet. You are having this one first sheet. Remember this one first sheet. Okay, first one. And sheet you are having chromosome. This. The third sheet you are having linkage map, which you have constructed just before this study, just before this QTL mapping exercise. Okay, then genotyping, and after that phenotyping. This is phenotyping data. Okay, now select this one and click on start. Sorry. Sorry, one step I missed. Okay. About result for the time being. So these are the parameters. Okay, parameters you have to set as per your will. So missing phenotype, what you want, whether you want to replace with the overall mean or you want to delete straight away. So I would recommend a delete for the timing, delete, and then mapping method. You can have multiple mapping method simultaneously. Like it is selected method is ICI mapping. You can have single mark analysis along with that also, and whatever you want to do. ठीक है. इसके बाद threshold, load threshold 3. You can put 3. Generally here it is 2.5. You can divide it 3. If you don't want to do it manually, you can go for permutation load threshold. What permutation do? It just means create once the QTL interval is identified or marker associated with QTL is identified, it reshuffle all the phenotype and see whether the QTL which has been detected after reshuffling the phenotype, whether it is detecting the same QTL or not. If it is detecting the same QTL, means there is problem because the whole phenotype has been shuffled. So if it is not, if in after 1,000 time it is not detecting any QTL, means it is means the load is means at p value of 0.05 is significant. But now you can put 3 means 1,000 time whatever linkage you are getting with the trait of interest QTL or that can be considered as our this QTL of QTL. So now you can go for start. Now I have changed the parameter, so there will be 2 results. Hello.
हेलो सर.
कोई क्वेरी है क्या अभी?
नहीं, अभी तो नहीं है.
ठीक है. So I think results has been there. So by double clicking, you can have all the information. These are the result files. So this is the QTL interval which the analysis have identified, and it has given additive information, phenotypic value explained, and load score as well. So it has identified located the QTL for the resistance at chromosome number 4 and chromosome number 11 with load score 1 QTL with a 5 load score, and phenotypic variance explained is 12.25. Now you can create a map by just click on F2B. Create. So this is the your linkage map. Hello. You want marker name in the left. You can change it to the right or left as per your will. Marker position in the left. Space type, it should be position. Okay. You can give load score along with this, and QTL also, where the QTL is there. You can play around with all these thing available with you. Okay. and you see chromosome number 4, 1 2 3 4 4 you can see here, RM4 dash underscore 62 and RM3 35. Within that region, the underscore Ilawa, if you can give QTL name also. You see QTL name. And if you want to change color, if you want to give centromere, you can give centromere like that. If you want to change color, change image style, chromosome, fill. So currently you can play around. You can change the color, and if you want to save the graphics, means you want to save it to clipboard, you can paste it anywhere, or if you want to save the image, you can save in different formats like JPEG, save. So these are the after means mapping. You can do anything. Okay, this is regarding QTL mapping exercise. Any question? If any question is there, you can ask. After that, I will start this genomic selection.
किसी को कुछ डाउट है तो पूछ सकते हैं आप.
If no doubt, then I can proceed.
सर कंटिन्यू सर.
एक सेकंड कॉल. Okay, next is genomic selection. So genomic selection is nothing means it's a new concept, but based on the older principle only. There is nothing means there is nothing especially new about it. Apart the information being the kind of information being utilized. Earlier we were doing selection based on phenotype and predicting the performance based on your heritability and breeding value. So there is only thing the information which we have at our hand that is based on marker information. So that is being utilized right now means in this scheme. So decision making based on the prediction using genome-wide markers, and selection on the genetic value predicted from marker could substantially increase the rate of genetic gain, because you are having a lot of information, just not the phenotype. You are having relatedness based on the SNP information, marker information. You are having co-ancestry. You don't have to go for co-ancestry, which we earlier used to have. Like we have to put in the matrix how much relationship is there, means, and that too calculation was very difficult at that point of time. Lot of calculation has to be taken over, and the availability of the software at that means I'm talking about it is 1990s or 1980s. Very less number of people were having access to those kind of software, and people used to go to them and calculate ancestry metrics and all. So it was very difficult at that time to go for means selection based on the phenotypic value and blood analysis and all. But now, as with the advancement of means computing facilities and development of different algorithms, it is becoming very easy. And information like SNP means genotyping platform, it has become very easy to identify what kind of relatedness among the lines are there. So this is the selection and genotypic value we can predict based on this means genotyping files, and we can predict the performance of the individuals. So this is the most famous breeder's equation. Uh, every breeder wants to have variety as soon as possible because it is competitive world. As soon as you are having your product at your hand, you can means monetize it as soon as possible. Okay, so I'm talking about this is the kind of competition private companies are having. They want to have thing means product in pipeline as soon as possible, so they are more efficient at these things. So this your delta G per year means genetic gain per year is dependent on your selection intensity, your accuracy of selection. This is where genomic selection works, because I is at your hand. If you want to have what kind of intensity you are putting in the population, and sigma A you cannot control. It is means you can control by adding variable kind of material in your population, and you can get it. But apart from that, you are relying on the natural variation only, and L is your generation interval. This can control by means now you are talking about rapid generation advancement, where shuttle breeding and facilities like greenhouse can be utilized to advance generation as soon as possible. So this gives a means a brief overview how the selection efficiency can be maximized and gain can be maximized. So you have to control means you have to control at least accuracy and variation. Variation by means creating variable population by crossing and incorporating the variable breeding material in your breeding program. So this is the basics. You all know about this average effect breeding value. So majorly in case of genomic selection, we are concerned about genomic estimated breeding value, which is calculated as the sum of the effect of dense genetic marker like SNPs or haplotype across the entire genome, thereby potentially capturing all the quantitative trait loci that contribute to variation in the trait. So here we are not concerning only single means whenever you go for QTL mapping, you are having a major QTL which is being utilized in your breeding program. But there are other genomic region which are contributing to the trait of interest. But the effect of that QTL is so minor that it cannot be utilized. Say for example, if you are having 4 QTL, okay, 4 QTL for a trait of interest. So how many population will it require to get a single homozygous plant having all the QTLs? So it will be very cumbersome to control 4 QTLs. Means uh, if you are talking about 4 QTLs, and it is very difficult out of I think 4 raised to the power 1 out of 256, at least 5 times of that 256 require to get at least 5. Okay, but there are minor effect QTLs overall which are contributing the trait of interest, which we are ignoring in case of QTL studies. So genomic selection came with that principle to how to utilize those things, how to capture those things as well. So basics. Okay, anyone you can tell what is the blue picture says? The graph in a blue picture, what does it say? So let me conclude it. Uh, it is saying that there is a complete coherence between breeding value and phenotype. Whatever the genotypic worth of an individual is there, it is reflected in terms of phenotype. And the slope of the line, that is B, means that is 1. So always means breeding students are having a confusion about why we are having regression coefficient B and when we are having heritability estimates. So in quantitative trait, B means H is having single formula, that is covariance between breeding value and phenotype upon VP. But B can be estimated from B is having B can be generalized in terms of many things. Means if you are taking into consideration a multi-environment, so here goes your covariance that has to be accounted for. So B can cover those things. So it is generally considered, means over year, if you want to have overall picture, how much gain we have got. So B can give you that picture. Means regression can give you that picture. Okay. Second one, you see there is 0.25 coherence, means unit change from 0 to 10 lead to change in the 0 to 40, means 1 divided by 4, that is 0.25. The heritability is 0.25. That is actually B. So this is the condition. This is the situation available at a breeder's hand. So, and it depends what he want to achieve. So for example, if you are having, if your trait is controlled by few QTLs, okay, so you can go for integration of those major QTL and then select. But if the trait is governed by lot of QTLs, number of loci are governing those traits, so in that case you cannot go for means integration. Integration will become very difficult, and selection of individuals with all those QTLs very, means it will require a lot of area, a lot of labor, and a lot of monetary, means monetary cost. So it is better that we go for genome-wide selection, identification of elite lines out of that. Okay, then this is a kind of very nice picture which has Bernardo et al. In 2008 has given, which shows what is your what should what institute or single lab or person can go for, until unless a lot of resources are there. It is a multi-institutional approach if you want to have a better result, because it requires robust phenotyping. Robust genotyping means hello,
कनेक्शन मिस हुआ था
इधर से तो नहीं था, मेरे तो आ रहा है.
ओके ओके अभी आपकी स्क्रीन शेयर हो रही है
Screen share का, अच्छा screen share, okay. Okay, so multi-institutional, and I'm telling you because it requires robust phenotyping, because if you are missing it at the phenotyping end, you will not land means anywhere. Phenotyping is very important aspect, whether you are doing it in genomics facilities or at a field, or it should be tested at least 4 locations to come out with the good results in case of genomic selection. Because the ultimate product of your selection is your variety, and it is going to be at field, and it should perform at that point of time. And if not working, means there will be an apprehension like genomic selection, a lot of resources are invested, but the results are not up to the mark. So when we are delving into such kind of exercise, it should be on the firm ground. So this is general scheme of the your genomic selection, where you are having a training population. Training population means a population. Training population means you are having individual which are being utilized in your breeding programs. and those training population has to be phenotype across the locations, and genotype as densely genotyped as well, and a model should be trained. Model should be trained like what is the genotypic change in the means genotypic contribution toward the phenotype, means like regression analysis is there, means different models are there, it's not like only regression, and based on that training model you are going to genotype your breeding material and calculate GEB based on the genomic estimated breeding value. You are going to go for selection. This is general outline. It is not means accurate one, but it's for better means understanding what is going on in genomic selection. So this is the actual working scheme. So I think most of the SAUs and institutes are having programs like dialyl mating designs, DSM and DSM designs, and lot of historical breeding progenies, and all those are having with them. So what can they do is, if you are at hand, that you can access, you have, means if you are having say for example 8 parents, how many dialyl possible combinations are there? 28 combinations are there, at least 8 into 7 divided by 2, so 28. So if each cross you are having at least 40 individuals, so it means you can maintain population size as per the resources available, and labor and whatever required is available, you can maintain that population size, and you use those population and fix that material to the homozygosity, and after that you go for the selection of the qualitative trait of interest, means it should be heuristic exercise, because just going directly to the genomic selection will not serve the purpose. At least you should select at earlier stages those for those traits which are qualitative in nature, because for selection based on those qualitative will be easy. So say for example you are having at that point of time 5,000 times, and out of selection from the selection based on qualitative trait you are coming up to 1,000 lines, then go for a station trial of these 1,000 lines, and select out of these 1,000 lines 200 lines and send them to multi-location testing. Okay, मल्टी-लोकेशन टेस्टिंग में एकदम एग्जॉस्टिव इनका फिनोटाइपिंग की do the phenotyping of these 200 lines across the location, and genotype these 200 lines densely, because you cannot genotype 1,000 lines, means you can do if you are having a lot of resources, you can do it, but in my opinion you do dense genotyping with these 200 lines and multi-location testing of these 200 lines, and as these are derived from these 1,000 lines, means coming out of these 100,000 lines which are having similar parents, because it is coming from the dialyl parental designs, so relatedness will be there. Okay, so based on these 200 lines you go and genotype the rest of the station trial lines. Okay, you have means this is your training population which has gone through multi-location testing, which has been highly genotyped, highly phenotyped. Now based on the model you select individuals from the station trial based on the GEBV, genomic estimated breeding value, and out of these 1,000 lines you come up with 40 or 50 lines. Okay, these 40 lines can go into varietal pipeline, and or if you want to have another cycle of selection you can utilize those to generate another breeding population, and you can incorporate uh, the other lines from outside of this breeding population, and this cycle goes on. Then it will become your marker assisted recurrent selection, means genomic selection of repeated cycles. This is the actual working scheme. If you want to have this you can have, I will share with this in the manual. And this is the data analysis framework what you are having: genotyping of whole station trial with a means at station trial you are genotyping whole population and you are creating a G matrix. G matrix is your related net matrix and ट्रेनिंग सेट सेलेक्ट करेंगे इसमें. अब ट्रेनिंग सेट को हम जो है मल्टीपल लोकेशन में मल्टी इवैल्यूएशन ट्रायल में डालेंगे. मल्टीपल इवैल्यूएशन ट्रायल डालने के बाद इसमें फिनोटाइपिक ब्लब करके, अगर सिंगल ट्रायल एनालिसिस करते हैं तो फिनोटाइपिक ब्लब कर लीजिए, डाटा क्लीनिंग करके इसको जो है जीनोमिक प्रेडिक्शन के उसमें यूज करेंगे. दिस विल बी आवर ट्रेनिंग पॉपुलेशन and then GEBV estimated from multi-location trial data. Your GBLUP model can be utilized for selection. So this is the general framework. Okay, this is with this I'll conclude. I can actually I don't have that kind of data set, so I cannot show you. I am having data, but it's not yet published, so it's not
No
good.
issues.
It's not good for me to show that. Otherwise, I can tell you about the models. If anyone is interested in kind of genomic selection in future, or if you are having resources at your hand, so my advice is to first have a theoretical background. Theoretical background of what is BLUP, basic best linear and biased predictor, what is BLUE. You have to go through these things. These all are mathematical, and I cannot explain this in means one hour exercise. This require a firm ground of mathematical knowledge. Okay, so if you are well familiar about matrix operations, then you go for GBLUP. What is GBLUP and what is the difference between the BLUP and GBLUP? So in BLUP earlier it was based on the ancestry correlation, co-ancestry based type. Based on the ancestry, you calculate the coefficient of ancestry 1/3, 1/4 like that. But in case of GBLUP, apart in place of that co-ancestry matrix, you use the information co-ancestry matrix derived from your genomic data. Okay, that genomic data is then used for your training of model with phenotype of multi-location trial of selected individuals. And one more point is that whenever you are going for development of your population, for training your training population, it should be related one where you have to go for selection. If it is diverse, the prediction accuracy will go down. Whenever there is more related individuals, whatever you are going for developing genomic selection training population, it should be related one. Okay, generally in textbook you see germplasm training population. It's not so. If you use germplasm as training population, then the kind of population where you are going for selection, that model will be totally different, because the kind of relationship with the training population and your breeding population is very less. So the chances of accuracy will go down. So accordingly, you have to very careful about your training population, development of training population. So I'll be saying all the best to you, all of you. Delve into these things, and if you means get opportunity to work such kind of project, it will be very nice. Okay, thank you, Vijay.
So thank you very much, sir. This session is open for discussion. So anyone having any doubt, sir is here, so you can ask.
So can you say again what is training population? Please
What you want to know about training population?
so the characteristics, what difference, how it is differ from the general genotypes or accessions.
No, it is no different. Actually, the breeding population you are having, it is very large, so you cannot phenotype all of them across the environment. Okay. Say for example, you are having a breeding population of 5,000 lines. Okay. If you are going for multi-location of 5,000 lines, how much time it will take, how much labor it will take, how much resources it will cut. You see, out of that 5,000 lines, you select 200, 300 representative lines, because it is coming from the means you have crossed in a particular fashion, and it is coming from that from that population breeding mating designs. So it will represent your this one your breeding population. select that subset of that training population or breeding population and use it as your mimic and circulate it all over environment where you are targeting for your product. And out of those and means I told you a phenotype in all those locations and train a model with p value at highest. Your regression should be highest, and based on that training model, you do select from your breeding population. You do genotyping in the breeding population also, but phenotyping you are only doing in training population. That is the major difference. Are you? Am I clear?
Yes, sir. Thank you, sir. Thank you.
Okay.
Anyone else? So thank you, Amarkant sir, for this wonderful lecture. And
Thank you, Vijay. And I'm sorry in between of interruption in between, because actually meeting is going on. That's why I was getting a lot of calls in between.
no issues, sir. Actually, this was very wonderful session, and you have covered all the things in very short time. That was very difficult. Actually, the topic is very large, but you managed to deliver it in within the time limit. So thank you so much, Amarkant sir, for joining us and
Vijay.
delivering your
Thank you,
lecture to our participants, sir.
Okay.
Hello, sir.
Mhm.
Sorry, sir. I
Yes.
have one more question, sir.
Okay.
Sir, there is a line in VD Singh and AK Singh, sir. Training population should accept low collinearity between marks. What is the meaning for that, sir?
What is
Training population should accept a low collinearity between markers.
low collinearity?
Between markers. Because low collinearity between marks lead to ये दिया सर, because high collinearity reduce the accuracy of GS model. हमें लो कोलिनियरिटी चाहिए सर ट्रेनिंग पॉपुलेशन में. अगर कोलिनियरिटी ज्यादा होगी बिटवीन मार्कर के अंदर, तो जीएस मॉडल की एक्यूरेसी जो है वो रिड्यूस हो जाएगी सर.
Okay, let me check. I cannot comment on this as such. अभी नहीं कर सकता मैं.
Okay, sir.
If this query, Vijay, you can write it if there is any means possible
Jaspal,
answer to this.
1 second. I think it is a case of multicollinearity, sir. I think so. So in multicollinearity, in whatever you model run, so whatever your explanatory variable means independent variable, if they are more correlated, then definitely it will reduce their model's accuracy.
यस सर, ये वाला क्वेश्चन भी आया था सर. अभी जो पीएचडी का जो एग्जाम हुआ था, एंट्रेंस एग्जाम, सर जोधपुर ने ही बनाया था सर वो, और ये लाइन दी हुई थी सर उसमें भी.
हाँ नहीं, मल्टीकोलिनियरिटी डेटा में होता ही है. मल्टीकोलिनियरिटी इसका मतलब ये होता है कि जब भी आप कोई डेटा लो ना, जैसे हम एक इंडिपेंडेंट डाटा लेते हैं और एक डिपेंडेंट डाटा. सपोज मेरा डिपेंडेंट डाटा है, यील्ड मेरा डिपेंडेंट डाटा है, ठीक है, और मल्टी डिपेंडेंट डाटा आपके हाइट वेट बहुत सारे हो सकते हैं. अब जो इंडिपेंडेंट डाटा है आपस में, वो आपस में कोरिलेटेड नहीं होने चाहिए आपस में बहुत ज्यादा, ठीक है ना. अगर वो केस आएगा तो मेरा मल्टीकोलिनिटी का केस आएगा. और जब भी मेरे डेटा आपस में इंडिपेंडेंट बहुत ज्यादा कोरिलेटेड रहेंगे, तो वो एक्यूरेसी मॉडल में जो आपके आर स्क्वायर वगैरह वो सारा का सारा डिप करेगा. और जब आर स्क्वायर डिप करेगा तो डेफिनेटली उसकी एक्यूरेसी आपकी लो जाएगी.
ओके सर.
I think it is clear now, but you can put that question in the manual if it is there,
Yes, sir.
and with probable answer.
Yes, sir.
Okay,
Yes,
okay, sir.
definitely.
Thank you. Thank you, sir.
Thank you.
So thank you, Amarkant sir, and thank you, Dr. Kapil also. And this session is over now. We will meet tomorrow at 11 a.m. Thank you everyone for joining us.